I would first like to thank the organizing committee giving me this opportunity to introduce and share the excitement of this new Earth-based electron pulsar, which enable completely new regime of ultrafast electron microscopy. I suspect for the most audience today, it is the first time to hear our company name Euclid Tech Labs. Indeed, Euclid is a newcomer to the field of electron microscopy. However, we are a well-recognized company in the field of particle accelerator community. Euclid was founded in 2001. It's a company specializing in development of advanced electron structure interaction apparatus for a variety of applications. Areas of expertise include electron accelerators, high-power components, photoinjectors, diamond-based electron sources, so on and so forth. We're still small up to present. We have 16 research staff, mostly host PhDs, 3 technicians, and 3 administrative staff. We have 2 main offices. The research and the lab facility is located in Bolingbrook, Illinois, which is a suburb of Chicago, headquartered in Washington, D.C. Historically, we have very tight collaboration with national labs and institutes. A couple years ago, we had opportunity sitting with a few scientists in a material science conference. During the conversation, we were inspired and encouraged to step into the field of electron microscope. 3 years later, there we are. We invented and demonstrated a laser-free electron pulsar which can retrofit any transmission electron microscopes. Also, in principle, the similar technology can be extended to SEMs, providing picosecond pulsed beam with unprecedented 10 GHz repetition rate. This work was selected at R&D 100 Award last year. Today's talk, I will first briefly describe how this gigahertz TM pulsar works. Then its main associated features, application range in terms of temporal and the spatial resolution, followed by a few application examples that we have done with collaborators. Will be finally concluded with a customer option. Up to date, when we talk about ultrafast or stroboscopic electron microscopy and the proper probe experiments, the first and maybe the only concept in our mind is a laser-based pulsar system, which is inevitably associated with a delicate and expensive laser system. Of course, it also requires a swap beam photocathode. Ultimately, the beam will be affected because intrinsically the best beam emittance is produced by a cold field emission cathode. Is there a way to produce ultrafast electron pulses without using photoemission? The answer is yes. To use a modern electron accelerator technology, we are able to manipulate the electron beam in 6-dimensional phase space. The basic concept is quite simple. Imagine you have a DC beam from a TM gun coming in. You let beam go through a space full of electromagnetic wave. The specialty comes from this electromagnetic wave has a field pattern which only kicks the beam transversely. The EM wave is time variant at certain frequency, depending on the arrival time of the beam. The electrons suggest dancing, or more scientific term, electrons are sinusoidally modulated. At the exit of the space, or after certain drifting distance, we can place a small aperture so that only electrons at the central line can pass through. The rest of the electrons will be blocked. After this aperture, the beam is chopped to pulses. The importance is the electrons on axis are experiencing minimal transverse cake, thus minimal effect on the beam emittance. Okay, now how to implement this idea? The first catch is to use a microwave cavity, operating it in deflecting mode. Researchers had demonstrated a few years ago. It works, but it has some issues. First, a cavity uses a fixed frequency, which means you can only obtain electron pulses at a fixed repetition rate, which may not be suitable to your sample. Second, the aperture has a finite size, so the electrons passing through the aperture has a certain wave, which means they still remain transverse momentum from a different cavity, although it is small. How to improve it? Instead of using microwave cavity, we use a special designed traveling wave strip-line kicker to modulate the beam. It is an ultra-broadband device. Operating frequency is determined by external RF drive, which is completely tunable up to 12 GHz. So, user can define whatever frequency matches to their study samples. Another uniqueness is that we introduce a second kicker called demodulator, which is operated out of phase of the first modulator to compensate the residual transverse momentum from this kicker 1. The RF drive to K1 and K2 are synchronized but independently phase and amplitude controlled. So K2 can be set to beam streaking mode, thus we can visually see the beam separation in time. The middle bottom picture is measured beamlet at the TM downstream phosphor using the streaking function of the demodulator. We do have another pulse picker technology, which can slow the pulse repetition rate from gigahertz to hertz level without sacrifice the electron pulse length. Limited by time, I won't talk in details. This slide shows two pulsars that were installed in two JOL machines. The pulsar in principle can be adapted to any TEM. We work with TM manufacturer to make sure the pulsar is compatible to TM vacuum and mechanical interface and radiation safe. During the installation, the TM gun will be lifted up. The pulsar is installed, then TM gun is moved back. Figure on the left shows how the pump probe stroboscopic experiments can be done. The pumping signal can be tapped from the drive with a controllable phase delay. The system also provides the standard 10 MHz or 100 MHz clock signal, which can be used to synchronize with other sample pumping sources, including a laser system to pump the sample if the user had already had a laser. There are many unique features of this tunable TM pulsar system. I just name a few important ones here. The first one is unbeatable high repetition rate. 12 GHz of the pulse rate has been demonstrated, which is a few order of magnitude faster than laser-based pulsar. In general, in a laser-based pulsar system, one electron pulse have more electrons because the commonly used photocathode has a large emission area. However, under certain circumstances, like a single electron imaging for radiation damage sensitive material, in order to obtain an image with the same signal-to-noise ratio, laser pulsar system require 100 or even 1,000 times longer time. To maintain the beam stability in such a long acquisition time is still very challenging, but it won't be the case for our gigahertz pulsar. The second feature I'd like to emphasize is ultra-wide tunability. It includes not only the tunability of the pulse rate, but also the tunability of the duty cycle. Or, in a simple word, the user can easily, through the provided GUI, change the electron pulse width from as short as 5 picosecond to 50 picosecond, or even to 100 nanosecond with optional module. The repetition rate can be seamlessly changed in gigahertz range or hertz to megahertz range depending on the selected module, so that user can have a choice of different combination of time resolution and rapid data collection suitable to different samples. The laser-free pulsar is a cost-effective option. This is understandable because the level of maintaining a laser system requires much more resources than sturdy RF electronics. At last, I'd like to mention this gigahertz pulsar is in principle can be retrofitted to any TEMs. We have had 3 installations for GEO TEM since our debut in 2019. We plan to make a pulsar for in-use FEI TEM starting from this fall. This slide is giving direct visualized timescale showing the basic temporal parameter space of laser and laser-free UEM. Here I'd like to make a few time-related concepts a little bit clear. When we talk about using ultrafast electron microscopy to study time-resolved ultrafast phenomena, we mean the electron pulse length has to be in the range of picosecond or femtosecond level, which provides picosecond or sub-picosecond probing temporal resolution. However, due to the Coulomb force, the number of electrons that can be packed in such a short time is limited, far less than a million, for energy of the hundreds keV beam. Therefore, in order to achieve picosecond or sub-picosecond time resolution, we have to use a stroboscopic mode. Doesn't matter if it is laser or laser-free approaches. In stroboscopic mode, the second important time-related concept is pulse repetition rate, which is determining how quick you can get one image if the number of electrons per pulse is in the similar level. The third concept is duty cycle, which is just a product of pulse length and the rep rate. As for the single-shot imaging, considering the required million electrons, the electron pulse length has to be in the range of the 10 nanosecond. If we like to reduce the pulse length into a sub-nanosecond level, MeV level energy beam is needed. Back to Euclid laser-free pulsar system. The basic model provides 5 picosecond to 50 picosecond tunable pulse length, with tunable pulse rate up to 12 GHz. With the pulsar picker upgrade option, we can extend the pulsar rate from Hz to MHz level, with still maintain the 10 picosecond pulse length. This option also provides the function of nanosecond pulse length for possible single-shot experiment. Therefore, this pulsar system can cover very broad spectrum of physics, chemistry, and life science phenomena. The demand from the life science is always a main driver advancing the TEM technologies, in particularly the rapid developed cryo-EM. Up to date, the spatial resolution of TEM or cryo-EM have already met most of the needs of biology research. Because Euclid TEM pulsar can retrofit in principle to any TEMs without changing the original cathode and column optics, you won't lose the spatial resolution. In addition, it can provide two more features that may help the life science research. The first one is reduced radiation damage in the cryo-EM if using pulsed beam. In recent years, a few papers had independently studied and confirmed that the pulsed beam may reduce the radiation damage of samples under the same accumulated dose. We also did a comprehensive study of the radiation damage using our gigahertz pulsar in a room temperature TEM and came to the same conclusion. We expect that the improvement factor for cryo-EM is even larger. I will show the results in a slide later. The second feature may be used to study the time-resolved biology structure dynamics. When people do stroboscopic pump and probe experiment, Euclid pulsar system can provide variety of pumping signals, like RF signal, fast electrical pulse, or standard clock signal to synchronize users' pumping signal generators. The large tunability and flexibility of the beam pulse timing structure gave users the most options of samples to be studied. This slide lists 4 different study cases as an example that Euclid laser-free pulsar may apply. Okay, now let's see a few examples of using gigahertz pulsar. The first example is a direct visualization of electromagnetic wave propagating in an interdigital structure, which is shown in the top right figure. The interdigital line is one of the basic building blocks for RF maps. Depending on the substrate, it can form surface acoustic wave devices, like the SAW filters that are used in every cell phone. It also can make biology or chemical sensors. Using Euclid gigahertz pulsar, scientists from Brookhaven National Laboratory and NIST did a pump and probe stroboscopic experiment on a 5 GHz interdigital line. In the experiment, the RF signal that is used to
drive the gigahertz pulsar was tapped off to pump the sample. It is the first time direct imaging of the effect of time-variant electromagnetic field in such a high frequency. Detailed results are presented in the reference shown at the bottom. The success of the study demonstrates the promise of inexpensive laser-free UEM in real space visualization of the dynamics in many research fields, especially in the electrodynamics in miniature information processing devices. Similarly, using pulsed beam and the RF wave excitation, instead of static images shown in the reference paper, now we can directly observe the spin wave propagation dynamics. These time-resolved images can more precisely characterize the process. Switch topic to the benefit of using gigahertz pulsar for radiation-sensitive material. This slide shows our latest effort on investigating the radiation damage reduction by using pulsed beam. This is an emerging field of study. Still a lot of argument around it. People usually argue it is material-dependent, energy-dependent, and some doubts to challenge the experiment method. In the study, two species were measured, paraffin and purple membrane. Both showed significant improvement in terms of radiation resistance under the pulsed beam. In other words, by using the pulsed beam, samples are more tolerable to high accumulated dose. The study also examined the fact of different duty cycle and different beam energy. The detailed results were published in an archive paper at the bottom. The dual-mode operation is another advantage of the Euclid laser-free pulsar solution. Once the pulsar column is installed, you can switch on and off by a simple click. Because the pulsar column is right after the TEM gun, the beam optic elements in the rest of columns remain the same. When the pulsar is switched off, the TEM just goes back to the normal operation mode. We have done the comparison before and after installation, and switch on and off the pulsar after the installation. As an R&D oriented company, we love to work with customers together, discussing specific need and requirement. Here we list some options we provide, but we are not limited. We understand every customer may have different scientific interests. As a new TM add-on product, we are learning how to explore and identify the real application of this laser-free gigahertz pulsar. We highly hope and expect that this device will leap forward the frontier of your scientific research. You are the customer we're looking for if you're looking for an ultrafast stroboscopy with highest spatial resolution available, or a retrofittable stroboscopic technology for picosecond performance that doesn't reduce the spatial resolution, and more.
To end this talk, I would like to thank your attendance. I also like to thank our funding agency collaborators during the development of this revolutionary product. We'll be very pleased to receive your email, phone call, and contact in person. We'll provide any assistance to realize your idea and imagination. Thank you.